Hello. Here we are on the last chapter for this week, and here we're talking about the control of microorganisms both in the environment and in the body. Okay. There was a lot of stuff in the last chapter, in chapter 8 in biotechnology. This chapter is a lot easier. Effectively, all the information can be dumped into two slides, and it is. And I'll show you which two slides at the end of this brief lecture. Okay. So the first thing that we're looking at is definitions in the control microorganisms. So I would encourage you to assimilate this terminology: what antisepsis means, what aseptic means, what cider-cidal means, what degerming means. Okay. Here's a definition. So if we're to look at aseptic, for example, that refers to the absence of microorganisms, pathogenic or otherwise. Cider-cidal, those suffixes implies the destruction of some type of a microbe. Okay. Here's one that's interesting: degerming. Okay. Degerming is one that's not common in our vocabulary. But when you go to a doctor and you get an injection of some sort, they take an isopropyl 1x1 pad and they degerm the area. Okay. And yes, the isopropyl alcohol is a germicidal and it's fantastic. But the fact that they're rubbing that on your skin means that they're actually carrying out degerming as well. And the definition of that is the removal of microbes by mechanical means. So now you know that when you get your skin clean with an alcohol prep, you're actually carrying out two different types of microbial control. Okay, one of them being degerming. So assimilate this terminology and what it means. Okay, it'll help you in the clinical environment, and some of this will help you on this week's quiz as well. Okay. What I want you to get out of this chapter primarily is the action of the antimicrobial agents. Each one of them has a different mode of action. Okay, it kills microbes in a different way. And I would like for you to correlate the microbial control with what their mode of action is. Okay. So, for example, one mode of action would be the breakdown of cell walls or membranes. Okay, when you have in particular with gram-positive organisms that have a tendency to live in hypertonic solutions, that cell wall is something that they depend on in order to maintain cellular integrity and to prevent themselves from lysing. But if you have something that whose mode of action is to alter or modify that cell wall, then they become susceptible to the osmotic pressures of the environment and they end up lysing. Okay, so that would be an example of a mode of action. Let's see. So antimicrobial agents, okay. We're going to talk about these agents more in depth pretty soon, okay. So we're just kind of touching on them a little bit this time. And so the reason I'm telling you this is because this happens to be one mode of action for antimicrobial agents. There's many, many more. But in this chapter, we're going to say that the mode of action for antimicrobial agents is to damage proteins and nucleic acids, okay? And logic dictates that if something is destroying nucleic acids, it is therefore destroying protein function, okay? So that's what causes the cell to die, is the fact that nucleic acids are being destroyed. Consequently, proteins are not being expressed and the cell can no longer be viable, okay? Heat-related methods. Clinically, this is important, but not with a patient, okay? This has to do with instrumentation and how to sanitize that instrumentation. So of all these, the one that we use in the laboratory the most is autoclaving. Okay, so autoclaving implies that you're taking media or instruments and you're sterilizing them. And the way that you do that is you bring it up to 121 Fahrenheit for 15 minutes. So high temperature, high heat, high humidity, and then for a good amount of time, 15 minutes. When that happens, just about everything dies, okay? There are exceptions. There's things that can survive autoclaving, if you can believe that. For example, there are some bacterial endospores that can survive autoclaving. Prions can survive autoclaving. So autoclaving is sterilization for day-to-day organisms that are susceptible to high heat, high temperature, and high moisture, okay? But this is typically what we use in the laboratory and in the clinic, okay? So here you go. Conditions, you don't need to remember these, but if you're in a clinical environment, you'll run into this. 121 degrees Celsius, 15 pounds per square inch for 15 minutes, okay? And this is what a clinical autoclave looks like. This is not what I have. Mine looks more like a canning pressure cooker, but it's still an autoclave. And effectively, in the academic, and at least in our academic environment, we can't afford this state-of-the-art autoclave. These things are incredibly expensive. But there you have it. That's what this is doing. You can see this person probably working at a dentist office because that's not a lot of room. So it's not like this person is autoclaving a lot of material. So probably at a dentist's office, they're putting all the instruments in there. And again, the conditions is that it's going to be heated to 121 degrees Celsius at 15 pounds per square inch for 15 minutes. You can see that all of that is digital, and when it's done, it cools off, okay? Here's another thing that you may find. You'll definitely find it in a micro lab or in a pathology lab, and it's what's called a HEPA hood. So the HEPA hood has a HEPA filter on the top, and it has what's called negative pressure. So as you're working on the microorganisms, this negative pressure is taking anything that comes out of your vials, and it pulls it up and into the HEPA filter, away from you and the environment, into safety. And it gets strapped into that HEPA filter where it does not cause any damage. We do have these. They're state-of-the-art. They're beautiful. It protects our microbiology students when they're working on bio-level 2 microorganisms. And I love them. Okay, it keeps us very, very safe. Okay, this is one of the main mechanisms for control of organisms. Again, typically gram-positive. Because, for example, on our skin flora, we have 2 gram-positive organisms, which is Staphylococcus aureus and Epidermidis. They both have a thick peptidoglycan cell wall. And because of that thick cell wall, again, they're not susceptible to the hypertonic environment that they're in, okay? But if you take antibiotics, one of the main functions of antibiotics is to inhibit the production of the peptidoglycan cell wall. It makes a cell wall tenuous, pliable, which is not desirable, and then the osmotic pressure is too much and the cell lyses, okay? So this is one of the main, incredibly, as trivial as it sounds, this is one of the main modes of bacterial control, is having them lyse due to a hypertonic environment through osmotic pressure. So if you don't remember this, you have to revisit your biology. Hypertonic, okay, means that there's more solute outside of the cell than inside the cell, okay? And because of that, if the cell's cell wall succumbs to the osmotic pressure, the water inside the bacterium will flow out in an effort to bring an equilibrium in the outside environment with the inside environment, and in doing this, it lyses, it breaks up. Okay. Let's see here. So let's look at the main protagonists, the main things that we use for control. Okay. So phenol and phenolics. Okay. So here's phenol. Okay, phenol is a phenyl group with a hydroxide. That suffix "ol" means that it's an alcohol. Okay, so these are all alcohols. What's an alcohol? It means it has an OH group, a hydroxide. Okay, so here's phenol. Here's bisphenolic, bis meaning two. Okay, and we used to use something similar to this in the clinical environment when I was in the Army. They used to call it Vesphine. This stuff is very, very useful. What it does is it denatures proteins and disrupts cell membranes. Okay, so an excellent, excellent way to disinfect items. The only caution is that sometimes this stuff can be carcinogenic. Okay. So I encourage you that if you are using phenols and phenolics, that you read the fine print. If it's carcinogenic, you can still use it, but you have to don gloves, wear a mask, put a garment on, and separate yourself from the phenol and phenolics. But I like them because they when they disrupt cell membranes or denature proteins, they effectively kill microorganisms very, very effectively. Okay, alcohols. Okay, same mode of action. Okay, so when I go into the microbiology lab, the first thing that I do is I take a big bottle of 90% isopropyl alcohol. I spray it on the counter. Okay, and what it does, the alcohol emulsifies plasma membranes, and it also denatures proteins. What does denature mean? It means that it unfolds the proteins. In order for a protein to function properly, it has to have a very specific three-dimensional shape. When you add alcohol, okay, and that hydroxyl group intervenes with the folding of that protein, and that protein unfolds, it denatures. It doesn't have the proper three-dimensional shape anymore, and that's it. The bacterium ceases to be viable, okay? So, again, alcohols denature proteins and disrupt cytoplasmic membranes because it emulsifies them. And I even like the smell of isopropyl alcohol, and I know it is doing a great job in cleaning the environment. Okay, halogens. Okay, so if you look at your periodic table, halogens is a column all the way to the right. So we're talking about chlorines and bromines, and you use it all the time. If you are in a doctor's clinic, you use betadine all the time. If you're in minor surgery, you use betadine. If you are cleaning wounds before surgery, you're using betadine. If you are if you're using a cystoscope and you look at the bladder, you're going to be using betadine to clean the urogenital area. This is one of the most highly used microbial controls out there, okay? And what does it do? It damages enzymes by unfolding them, by denaturizing them. Very, very, very neat, okay? This probably looks familiar to you. So here's somebody probably with a wound of some sort, and they need to be scrubbed with betadine with a halogen in preparation to minor surgery or major surgery. I'm not convinced what's going on. I know what's going on here. Oxidizing agents, peroxides, ozone, all these. Mode of action, destroy microbial enzymes. Yes, that's true. But if you also remember the toxic species of oxygen, you remember that if you have relevant pathogenic microorganisms. They are typically obligate anaerobes, like Clostridia, and these guys do not fare well in an oxygen environment. So oxygen, peroxides, all of these provide toxic oxygen species that effectively kill obligate anaerobes. And this is a very desirable outcome, okay? Surfactants, okay? This is a topic that I find amusing because people believe that surfactants sterilize material, and that is not true, okay? Why am I telling you this? When you're at home and you're washing the dishes with detergent, that detergent is primarily a surfactant. And what a surfactant is a wetting agent. It is something that emulsifies lipids. And does it get rid of fats? Does it get rid of membranes? Yes, but it does not kill anything. A surfactant is an emulsifier and is a wetting agent, okay? If you are in professional food industry, okay, you'll remember the 3-sink way of washing dishes, and one of those sinks is a disinfectant. And that's because in professional food handling, they become aware, or they teach, that a detergent is not enough, because a detergent, again, a surfactant, all it does is get rid of lipids and nothing else. You need something to kill the residue microorganisms that remain on that plate or that spoon, okay? So you need to have another tank besides the detergent. So this is an important point to know. Now, are surfactants useful? Absolutely, okay? If you have a disinfectant and you add a surfactant to it, the end effect is going to be a multiplication, okay? So this means it's going to be a synergistic outcome. So that means that whatever the effects of the original microbial control is, the surfactant is going to multiply it, because it's going to allow it to enter or traverse or evade the problems associated with membranes and lipids and anything that is hydrophobic in nature. So very, very useful. Quats are very cool. This is a new type of surfactant that's come out. And not surprisingly, it deals with lipids and the emulsification or disruption of cellular membranes, okay? They are they're very popular these days. And this is what a quat looks like, okay? Again, is a detergent. How do you know that's a detergent? Well, a detergent has to have both hydrophobic and hydrophilic character. It has to have hydrophobic character so it can associate with the lipid that you're trying to get rid of. And then it has to have hydrophilic character so it can associate with the water that you're using to wash that lipid off. So any surfactant, any detergent, any wetting agent is going to have both hydrophobic and hydrophilic character. If you look at this, look at that long hydrophobic character. See, hydrophobic tail. And what that does is it allows this quat to associate with lipids. And see that little charged nitrogen, okay? That little cation is the hydrophilic, the part that associates with the water, okay? And here's another one, okay? Hydrophobic table, I can't even talk anymore. A hydrophobic tail with a hydrophilic head. All detergents have the same traits. Heavy metals, you guys just wrote a summary dealing with this. This is why you had to write this summary, because we're dealing with this chapter on microbial control and heavy metals. we were talking about using heavy metals prophylactically in order to save the eyesight of infants from mothers that happen to have N-gonorrhea or gonorrhea. Okay, so here it is again. Okay, 1% silver nitrate being used prophylactically on the infant, and again, it saves the infant from a life of blindness. Okay, here's another beautiful use of heavy metals, Silvadene. I remember using Silvadene a lot. I don't know why, but in our emergency room, we ran into a lot of burns. Okay, this is hard to deal with. First, second, and third degree burns, and we had to irrigate them to the very best that we could. A lot of times we used betadine. Sometimes we had to use local or general anesthesia to sedate the patient because this was hell on earth. It was very painful. And then once the burn was irrigated and cleansed, we would put Silvadene, and we would put 4x4s on top of it and wrap it loosely, okay? And if the area was big enough, maybe transfer of a germ layer transfer for skin growth, okay? But typically our burns were sufficiently small that they started to regenerate skin on their own. So I have used Silvadene topically for burns many times in the clinical environment. Okay, here's an example of a heavy metal that the author's using to show that there's an area here where the bacteria is not growing. So the heavy metal inhibits the growth of bacteria. Here's aldehydes, where an alcohol has an OH group, an aldehyde has a CHO group, okay? And what you need to know, again, is the mode of action. And what it does is it denatures proteins and it inactivates nucleic acids, okay? So once again, this inhibits the expression of proteins because if the nucleic acids are broken up, that cell is not going to be able to produce any protein, okay? Enzymes. This is a new, fascinating sphere of microbial growth, okay? Again, we dealt this in the summary, okay? So this was an opportunity for you guys to read and become aware of this new research area. And if you guys remember, the topic had to do with prions, okay? And as I said earlier, prions, which are what cause Jacobs-Creutzfeldt disease and mad cow disease in cows, you can't get rid of it. You can't use heavy metals, nothing will happen. You can't use halides, nothing will happen. You can't use alcohol, you can't use aldehydes, you can autoclave it, nothing will happen. You can irradiate it, nothing will happen. This thing seems to be impermeable to anything, right? So just about the time that we're losing hope, cue in enzymes. Beautiful. So now we have a prionzyme that can break down prions on medical instruments. And now, as you know, we're trying to do clinical studies for people as well. But this is something that we cannot destroy because a prion is not even an organism. It's a misfolded protein. We simply had no way of getting rid of this thing outside of outright incineration, burning it, okay? And now, thanks to enzymes, we can actually break down the prion and impede contamination and hopefully soon resolve Jacobs-Creutzfeldt, and even mad cow disease, which is incredible. So enzymes win where nothing else will work, okay? We have lysozyme in our tears, okay? So one of the things that we teach in microbiology, for example, is that we're always touching things with our hands. And then next thing you touch your face with your hands, and that's a means of introduction bacteria into your body, specifically through the eyes, nose, and mouth, okay? So if you contemplate how much garbage we touch with our hands, and then how often we touch our faces afterwards without thinking about it, it's a miracle that we're not walking around sick every day of our lives. And the reason that we're not doing that is because of this beautiful lysozyme. So when we deposit this bacteria on our eyes, the very tears in our eyes digest the peptidoglycan cell wall in these gram-positive organisms and destroys them by the subsequent osmotic pressures on it. That's incredible, okay? And I remember reading this a long, long time ago and thinking, I'm sure somebody has this, right? But I've actually since then come up on a protocol where I had to lyse gram-positive bacteria. And what was I putting in it in order to lyse it? Lysozyme. That convinced me. This stuff that we produce does in fact kill bacteria. Very incredible. So enzymes are fantastic. Antimicrobial drugs, these are interesting. One of the things that you should know, again, is antimicrobial drugs are naturally produced by microorganisms. Okay, and then we harvest these antimicrobial drugs for ourselves to protect ourselves against these microorganisms. So sometimes they come from bacteria. The majority of the time they come from fungi, like penicillin comes from a fungi. Okay, but it's interesting that we're using the same metabolism of microorganisms to defend ourselves from them. Okay, and here's our table bringing all that together. I told you that at the end of this little lecturette, I would show you the two tables where everything would be coalesced into one point so you can find everything in one place. This is one of those tables, table 9.5, and the other one is the one that we began on, which is this one. No, just lied to you. And the other one is this one that has the terminology of microbial control. That's it for this chapter. I hope you're having a wonderful day. Have a good morning, and I'll be uploading the laboratory here in just a second, okay? Have a wonderful morning.